all kinds of both family also have some exception. For example, coronavirus, COVID-19, it is the amylovirus, but it is resistant to detergent and it can be dried out. And the coronavirus can also be transmitted through oral fecal pathway. So if any of you have been infected by COVID-19, I believe you should have diarrhea syndrome, and all your toilet should have. If you are interested, you can swab the toilet. Should be lots of COVID-19, SARS-CoV-2. I think we start at 3:50. I remember it should be finished in 3:45 or 3:50. Anyway, we don't have break. So the third meaning important thing of the virus is the viruses usually utilize their genome very, very efficiently. Because I don't know your background, if you are the undergraduate student or the even PhD, you should at least have some idea on called genetic cloning. So for example, here's the genome of HPV, human papillomavirus that can cause cervical cancer. And just within this 8 kilo base pair double-strand viral genome, it can already create 1, 2, 3, 4, 5, 6, 8 viral protein. It is quite amazing. If you have any experience in constructing a plasmid, usually one plasmid, if you can have 1 or 2 genes, already amazing. For this virus, they just use the same amount of genetic coding area. They can express 9 protein. And you can see that some of the protein are even overlaid. So they use very, very short region to express as many protein as possible to make them simple. Use the smallest genetic material and produce the most of the thing. And of course, all the viral protein, if you want to have drug, you need to identify each protein function. So even here's a small virus, and I show you another big virus called Epstein-Barr virus. Its genome is 170 kilobase pair. It's nearly 20% of bacteria, so it's a very giant virus. But believe it or not, nearly all the gene segments of this virus also encode the protein, and all of them are actively expressed in the host cells. So it means the virus use their genome quite wisely anyway. And a virus, if they keep this genome or keep this viral protein, definitely have reason, or they will be quickly, or this virus keep this extra viral protein will quickly disappear. And I will use mathematical calculation to tell you why later, okay? Because viral evolve very fast. And what is the virus life cycle? So if you want to study a virus life cycle in the infected cells, we usually study these steps. What is the receptor for the virus? Because the first thing is the virus need to find the cells having the receptor that recognize the most out layer of the virus envelope or capsid, and that allow the virus attach to the cell surface and therefore go to entry. For example, receptor of COVID-19 SARS-CoV-2 is ACE2. So ACE2 can bind to the spike protein of the SARS-CoV-2. So SARS-CoV-2's cellular receptor is ACE2. It helps the SARS-CoV-2 dock to the effective host cell. So after the viral receptor and viral glycoprotein binding, and therefore the virus on the cell surface, and therefore perceived entry. It can either be performed by fusion or endocytosis, depends on different viral family. And therefore, after entering into the cells, the virus will release its genome and start to do viral protein transcription, translation, DNA transcription with RNA virus translation. And when they produce viral protein, they will hijack the cellular machinery to make them produce more viral protein and more viral genetic material, and therefore package into the new virus and release. And there are many, of course, more detailed mechanisms. But the principle is binding, entry, replicate, and release. So why we need to study biology for more detail? The basic biology is or how to say, molecular biology is actually to study a virus life cycle in the infected host, means to study every single step. Why it is important? Because if you block any single step of the virus life cycle, theoretically, you can cure the disease. For example, the vaccine we use for SARS-CoV-2, COVID-19 is actually the vaccines that contain this spike protein. So it allows the people, our body to generate a spike antibody. So after the virus caught with the spike antibody, and therefore this spike protein is not allowed to bind to the ACE2, so it's not allowed to enter the cells. Of course, we can also find some drug that block viral replication. So even if bind enter, but if it cannot replicate, it cannot release more babies to infect more cells, so can seize the disease. So if the biologists are fully understand each virus, this life cycle, theoretically, we can cure lots of virus-associated disease. But it's far more difficult than you imagine. I think, let me see how many slides we still have. Yeah, I think we just at the middle. So on the schedule, yeah. So next week we will finish another 50%. So maybe in the end I show you one thing quite interesting. And why the virus get mutation so fast? It depends on the error rate of the polymerase. For example, RNA virus, if they want to replicate, they need to use a viral RNA-dependent RNA polymerase. And its error rate at average is 1 mutation per 10 to 4 copies. COVID-19 genome is 4 times 10 to 4. So by using mathematical calculation, every single virus it generates have average 3 to 4 mutation. Okay. And each infected cell they generate 0.1 to 1 million mutation. So what I want to say is the mutation of the virus happen every single minute, single hours. Always new virus came out. Some of them have more advantage, some of them have less advantage. But after a long time enough, the best one will survive. So this is how the COVID-19 genome get evolved. At the beginning, 2019 December, there's only one strain called Wuhan strain. And just after 3 months, you can see that COVID-19 already has such many different family. And here is after 5 months, already have some major family. I think some of them are alpha chain and beta chain and gamma chain. And currently the computer cannot run. Yeah. So I just want to tell you how fast the virus can get evolution. So not so surprised there will be a new coronavirus totally escape from the vaccine you have already vaccinated according to their mutation speed. So we stop here. Let me make a note. We are here. We are here. Yeah. So we start from page 75 next week. Any questions? Question? If no more question, then we stop now. So see you next week. See you next week. Bye. Be safe. Nobody here.